We have Kelsy Inman-Carter, and she was working with Sarah for the summer.
So, hi, I'm Kelsey, and I was working with Dr. Sarah Hamsher this summer. And my project that might continue into the school year was focused on the epiphytic diatom communities of an invasive macroalga and its native relative in two Zonger mouth lakes here in Michigan. So, the purpose of my project this summer was to compare the epiphytic diatom communities of Starry, Stonewort, and Kara, and that's hopefully going to help us gain a better understanding of the alga-host epiphytic diatom relationship in freshwater environments, as well as provide us a comparison between the epiphytic diatom communities of invasive species like Starry, and native species like Kara. So these pictures here show some examples of the macroalgae that we worked with this summer. Over on the left is Kara, and then on the right is Starry. So they can look kind of similar, but some of the differences are that Kara can appear more bristly, while Starry is smoother. And then Starry also has these star-shaped bubbles right here, and has unequal branching, which can give it like a less symmetrical look than Kara has. My project itself was focused on the epiphytic diatoms that grow on these macroalgae. So epiphytes are non-parasitic photosynthetic organisms that grow in other algae or plants. And even though they're not parasitic, they can still affect the host of the plants, either positively by increasing the organic compounds and CO2 that are found in the environment, or negatively by increasing predation rates, or even possibly like blocking light from reaching the host. So diatoms are algae, are microalgae, and they come in lots of different shapes and sizes. Like lots of other types of algae, they can be either singular or in groups. And this one over here shows one that I collected this past school year for my freshwater algae class on the Onda Public Schools campus. And they're really important primary producers in their environment and can consider comparable or contribute, comparable productivity to the phytoplankton that are found in the environment. So the plan this summer was basically to do DNA extractions and PCRs. When I first came into the project, I already had 80 samples waiting for me, courtesy of collection done last year, weekly through July, September by Dr. Hamsher's team. And then in June of this year, I went out again to collect some fresh samples with Dr. Hamsher, Sarah, Brooke, and Emily. So we had 2 sites for collection, one in Stonewort Lake and then one in Muskegon Lake. And at each site, we collected 5 samples of each of the plants. And alongside collecting Starry and Kara, we also collected samples of Valsmeria americana, which is sometimes referred to as water celery, and Conliget zostriformis, which is a pondweed. And aside from the plant samples, we also took 5 sediment samples and 5 plankton toes in each of the locations. And then after this first outing to gather some more samples, I went out and worked 4 times into the field this summer. The plan with the samples was to first extract the DNA, and we wanted to hopefully have 3 successfully extracted samples from each group of 5 that we had. And the way to test this was we used ready-to-go PCR beads for the first round of PCRs. And this was just to let us know if we needed to do any more extractions or even possibly gather more samples. And once we had samples all extracted, we planned to use the Kappa Hotstart PCR kit in order to amplify the metabarcode region, which for the purposes of our project was the RBC algae, and then sequence the metabarcode region using MySeq. As for the extractions this summer, we did 133 extractions using the Being Easy Power Soil Pro Kit, and that includes all of the algae samples as well as the sediment samples and plankton toes. And 85 of those were found to successfully extract diatom DNA, which is about a 64% success rate. Early on in the project, we did some sequencing on a couple of the samples from last year, trying just to give us an idea of what to look forward to going into the project. The first round of sequencing was done on 2 samples of Starry from Stonewort Lake, and all we found there was a non-native species, and that was done using primers that we aren't using currently. And then we redid sequencing with primers that we are using now, and we did 2 samples of Starry and then 2 samples of Kara, all from Pentwater Lake as well. And the 2 Starry samples were found to have the Amphibian species again, but one of them also had the Epiphytic species. And then the Kara samples, one of them didn't work, but the other one had a Symboplura species found on it. Moving forward into the fall, we plan to work with amplifying the metabarcode region using that Kappa hotspot PCR kit, and then work to sequence that metabarcode region using MySeq. And it's our hope that we're going to find a difference in the epiphytic communities, either based on the lake that the sample was taken in or the algae the sample was found on. And then as a wrap up, I just wanted to do some thank yous. So I wanted to thank the SQ program at Grand Valley for actually funding our project this summer and giving me the opportunity. I want to thank AWRI as a whole, and especially Dr. Partridge for letting me work in her lab this summer. And I wanted to thank Dr. Hamsher for guiding me through my first research project and just helping me whenever I had any questions. And then finally, I wanted to thank Brooke and Emily for just welcoming me onto their team and helping me feel really welcome this summer.
Kelsey, do you think the epiphytes are affecting the Starry at all negatively?
We aren't sure yet because we haven't really seen what is on each of the plants yet. We haven't gotten a chance to look at what's actually found on them. But it is seen in some other studies that the epiphytes can affect the host, so it's really interesting to look at.
I haven't looked at the data yet, but do you predict that there might be a difference in the epiphyte community between the 2 sampling locations between Pentwater and Muskegon?
So that is part of the big question, is we're trying to figure out whether it's like the water chemistry or the plants that are the difference. But in some of the literature that I've seen, it does seem like the water chemistry plays a role. So I think it would be interesting to see if there's a difference.
Do you know if that Kira's parasites release the
Yeah, so that we haven't discussed that yet because we wanted to see what came back using MySeq as compared to Blast first. But that is something we might have to look into.
Any more questions? Okay.